Welcome, Brookings Bio students. This is another installment in our quest through the cell parts chapter. Lysosomes is the next part we're going to learn about. And the word lysis has to do with breaking things apart. And if you remember back in our molecule chapter, we learned about dehydration synthesis, removing a water molecule to make a bond. And the opposite of that, we learned about hydrolysis, splitting with using water, adding those water molecules back to break the bond apart. The lysis part of lysosomes comes from that same root word. It means to break apart. And these are cell parts that are going to have digestive enzymes contained in small sacs to break down things in the cell that the cell would like to get rid of. Could be food particles being broken down in a single-celled pond organism of some sort. Think an amoeba, for example. It could be molecules that the cell doesn't want, old cell parts, organelles that have broken down. Maybe a mitochondria is not working like it's supposed to. Can be whole cells, like a bacterial cell. Can be cells with viruses in them. Anything the cell wants to break down can use those digestive enzymes in lysosomes to do that. If you look at how that works, for example, this is a little picture showing the edge of a cell here, and you have a food particle outside the cell, and we're going to bring that food particle into the cell and close it in a little compartment storage sac. And then we're going to add those lysosomal enzymes, those digestive enzymes, into the storage sac to break down the food molecules that are in there. We're going to watch a quick little animation that shows this happening inside of a cell. So here's my cell. Notice eukaryotic cell. It's got a nucleus, has some rough ER. Golgi bodies, and now we're gonna, maybe this is a little pond organism of some kind. We're gonna watch what happens when this organism reaches out and brings in some little pieces of food. We're gonna dump our lysosome containing those digestive enzymes onto that container holding the food. We're gonna absorb the food molecules that are nutrients, and then kick out our garbage. And lysosomal enzymes are going to be the digesters that help to break down those food molecules. Doesn't have to be food. It can be anything that the cell would like to get rid of. Damaged cell parts, any other sort of thing that the cell...
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. These enzymes digest particles or cells taken into the cell by phagocytosis. They also digest old organelles such as mitochondria. The hydrolytic enzymes that degrade proteins, nucleic acids, lipids, and carbohydrates are formed in the endoplasmic reticulum and then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as defective or worn-out mitochondria. This results in the destruction and recycling of these structures.
Okay, so let's go back to our PowerPoint here and see if we can talk a little more about some other instances where we use lysosomes. And one of those very important processes is a process called apoptosis, programmed cell death, the death of the cell that is programmed genetically into the cell, maybe as part of development or anything that is a way to maintain body cells, maintain the health of a cell. If a cell recognizes that there's something wrong with it, maybe a mutation has happened, maybe it picked up a virus or a bacteria of some sort. If the cell recognizes that it is sick, the lysosomes will actually do a self-digest on the cell and destroy it. And that is that apoptosis, sometimes called cell suicide. for the good of the organism. A place that we see this happening is in embryonic development. If you look at the picture on the left, it shows how your hands and feet form in the embryo, in the very early embryo. We don't actually see individual fingers and toes like in the second picture. The arm and leg buds actually form as little paddles. They look like ping pong paddles. And the finger bones are forming inside, but we don't see individual fingers and toes. And those come later as the cells that are in between the fingers undergo apoptosis, programmed cell death, to remove those cells and allow the fingers to be separated, and we start to see little fingers and toes in our embryo. At one time, you had a tail as an embryonic, as an early embryo. We have tails. And those also are removed with using apoptosis, self-digesting of those cells to remove them before the baby is born. Sometimes that something goes wrong with that message signaling system, and babies are born with little tiny tails, small tails that would be removed surgically. But it's part of that programmed cell death. The cell recognizes that it needs to disappear, and using its lysosomes to self-digest. Plays an important role in embryo development, as we just said, maintenance of normal body cells. As with cells get used and worn out, we replace those and destroy the old worn out cells. It's important in immune system responses. The white blood cells that engulf and take over bacteria or viruses use that same lysosomes to break down those cells and actually self-destruct once the viruses are inside to help your immune system fight germs. It's one of the problems that goes wrong with a cancer cell. Most cells that are sick recognize the signal that says there's something wrong and will do that self-digest, destroy themselves for the good of the organism. Cancer cells do not respond to those signals. Even though the cell is sick, it does not respond to the self-destruct signal. It's important in AIDS infections, in transplant rejections, lots of places where we need the cell to recognize correctly that something is wrong and use that apoptosis to self-destruct. So let's put some functions down for lysosomes. They're sacs containing digestive enzymes in very acidic conditions. Think about what would be in your stomach to break down food molecules. We have acid in there, hydrochloric acid, and there's digestive enzymes that work to help to break down those food molecules in your stomach. The function of the lysosomes is to digest anything that the cell does not want. Could be a food molecule, unwanted cell parts, broken mitochondria, for example, other kinds of cells, viruses inside cells. And those lysosomes play an important role as part of that programmed cell death, maintaining the health of the organism through apoptosis, cell suicide, cells recognizing that they are not well or not supposed to be there, and doing that self-destruct thing. Okay, so let's see if we can find some lysosomes in our cell parts picture. So we're going back to our cell parts picture, and I'm going to ask you to pick another color now. We're going to use another color for cell parts. And this part is, we're going to start in our animal cell. And if you look here in this cell, over on the left hand, right towards the bottom, there's a little structure. It looks kind of like a ring with smaller little sacs attached to it. That ring is a digestive compartment. There's something stored in there that the cell would like to break down or digest. Maybe it's a cell part, maybe it's molecules, maybe it's a virus, maybe it's something, anything that the cell would want to break down. It puts it in a little container called a vesicle or a vacuole, and then it dumps those lysosomal enzymes on it. So the little ring structure is not our lysosome, but those little sacs that are attached to the ring are the lysosomes. So we're going to color those in. And if you look in your animal cell, you'll see a couple other places where we see those little ring structures, those little storage compartments or or compartments holding something, and the attached little sacs are the lysosomes. They're bringing digestive enzymes to dump into that container and break down whatever is inside. So we're going to color just the little sacs that are attached to that storage sac compartment. Those are lysosomes. And lysosomes can also be free in the cell as they come from the Golgi where they're packaged and move throughout the cell to go somewhere to break down something. You can see some kind of just free lysosomes. Plants also have lysosomes. There was some discussion for a while about whether or not that was true, but we do think now that plants do have lysosomes, and we can see a couple here in the plant cell that are waiting to be digesters for something that the plant wants to do. You don't have to fill in every single one. Okay, let's put our name label in, lysosome. Notice plants and animals, eukaryotic cells, have lysosomes. They are membrane sacs containing those enzymes. And bacteria, remember, do not have organelles made out of membranes, so no lysosomes. Does that mean they can't break down things? No, they can. They would make and use their hydrolytic enzymes and get rid of them as they are needed, and they don't actually put them into little storage sacs like we do. Okay, we're going to go back to our flip book in our talk about our next cell part, which are actually two very similar related parts, which are the cilia and flagella. Notice vowels at the end. This is the plural word of flagellum and cilium, the singular. Usually often more than one together, so we usually use the word flagella or cilia. And the pattern that we see in these cell parts, they're made out of proteins called microtubules. We saw those in our cytoskeleton. We saw them in centrioles. They make the spindle fibers for dividing cells. Those microtubules come in pretty handy. And what makes them different now from the other structures we've seen with microtubules is this 9+2 pattern. If you look at how they are arranged, the microtubules are grouped in little, we call them doublets, little twos, around a center, kind of like a wheel, spokes on a wheel. And we have 9 little doubles on the outside and 2 in the center. That's the 9+2 pattern that we see. And both cilia and flagella show that 9+2 pattern. If you slice them in looking on the end and the cross section, you see that arrangement of microtubules. Flagella we have seen in, for example, a sperm tail that helps the little sperm to swim. Both of these structures are involved in helping the cell move in some way or moving something past a cell. Cilia we've seen in like little pond critters. They help something to swim or move a cell itself. Or in multicellular organisms, we see cilia actually attached on the outside of cells that are part of a bigger structure, a bigger tissue or a bigger organ. And for example, respiratory system in humans has cilia lining, the cells that line our respiratory tract have cilia on to help brush and move the dirt and dust particles that we breathe in, help to move them back up, out, and out of our respiratory system. And those little hairs on the outside of cells are the cilia. So what's the difference between the two? They're both made out of microtubules. They both have that 9+2 pattern. Basically, the difference is how many there are and how long they are. So cilia, usually lots of them, many short hair-like structures on the surface, and very, very short, kind of short and stubby little hairs. Flagella, fewer, maybe 1 or 2 together, and very long. And that's really the only difference, is the size and shape of those 9+2 structures. Okay, so let's get something in our cell book here. Made of microtubules. That would be a good test question. Tell me some cell parts that are made out of microtubules. They're organized in a 9+2 pattern, and that's what makes them different from, remember, our centrioles had microtubules, but they're kind of organized in a cylinder in little groups of 3s. Their job is to help move something in the cell, either outside, attached to the outside of the cell to help it move the cell itself. Many short are the cilia, function in moving cells, whole cells, or moving something past a cell. Think your respiratory system. Fallopian tubes, your oviducts, have cilia in them to help move the eggs along. We see that in a lot of body systems have cilia to help move something. Flagella, difference between cilia and flagella is, remember, size and numbers. Few and long hair-like structures, and their job is to move cells. Think sperm tail. I'm going to ask you to take your little pencil, and we're going to draw a little sperm here. Here's my head and tail, right? That's my flagella. Cilia, let's make a little cell here. Layer of cells with cilia, little hair-like structures to move something past a cell. Let's give it nucleus. Either move something past a cell or actually can be attached on the surface of a cell to help the cell to swim and move. Think some different kinds of little single-celled pond critters that might move with cilia. That is the end of our talk about cilia. We're not going to actually draw cilia and flagella on our cell parts picture. Not all cells have cilia and flagella, but you should be aware of what they look like and where we might find them and what their function is. So thanks for watching.